was feeling sort of unscripted here, so decided what I wanted to do is to kind of give you guys a perspective or framework to kind of go by as we continue through the rest of this unit on cancer, okay? Because where we left it off going in on today is now you guys have seen how a typical RTK pathway works from start to finish, and where all of the steps that are involved in activating a downstream step that ultimately leads to proliferation. There's always a negative feedback loop that works in there to try and keep it from being propagated. But basically now you guys know how everybody in that pathway works and what they're basically doing to cause cells to proliferate normally, okay? Well, now we're going to go back, and today and into Monday, and we're going to go through the pathway again. Only now what we're going to do is take a look at different places in the pathway where you can generate an oncogene, and that is you take a component that has a normal role in normal proliferation, and we're going to mutate it. And the mutation is now going to cause it to function as an oncogene. And now we're going to get abnormal cellular proliferation. And as you're going to see, as we go through the pathways, you can see up there, there's many, many, many places and many ways this can happen. And so one of the things now, before we get into that, is to understand what is happening to a cell and what are the pressures it's subjected to that cause this to happen. Why does it happen? Okay? So the first concept I want to get out there is this notion of oncogene addiction. Okay? And what it literally means is that a particular cancer cell at a particular time and a particular place, and you'll see where, the cells are always being exposed to some sort of external selective pressure. Okay? So for example, the ability to proliferate in response to an extracellular growth factor, like we talked about, that's a selective pressure. You only proliferate if the growth factor is out there and you can bind to it. So there's selective pressure. So if you acquire an oncogene that allows you to bypass that requirement, to be able to proliferate in the absence of a growth factor, those cells are now addicted to the oncogene that allows that to happen, meaning addicted that they can only proliferate if they have that oncogene. Take out that oncogene and now that cell can no longer proliferate. It's just like everybody else was. Okay? So we've got oncogene addiction, and that's what gives rise to what we call a cancer hallmark. And I'll be posting a review article you guys can take a look at today, where a couple of investigators went through and they basically identified what are constituting cancer hallmarks, features of cells that are unique to a cancerous phenotype. And one of them would be sustained proliferation. That's a hallmark, okay? So, for example, the hallmark there, if you had an altered RTK that would allow you to proliferate without binding to a growth factor, that's a hallmark, okay? Another hallmark that you're going to see would be you have altered metabolism. So now the cancer cell's metabolism doesn't follow what normal cells do. And that's a unique property, enabling. Or, for example, a cancer hallmark, as you're going to see, could be they overexpress GLUT1 transporters relative to a normal cell. Okay? They have more GLUT1 transporters. That means that they can basically import more glucose than a normal cell for metabolic purposes and sustained growth. Okay? Well, that also gives you an enabling characteristic. And that means that that cell, an enabling characteristic means that cell can now do something because of that hallmark. It means it can proliferate under metabolic conditions that normal cells can't. Okay? So the key thing to understand, firstly, is that oncogene addiction is going to be a genotype. It's a change in a gene. And that's going to come about due to adaptive mutations. You're applying selective pressure on a cancer cell, you're selecting for mutations that allow it to survive or bypass whatever that pressure is. Okay, it's just Darwinian selection. The hallmarks and the characteristics are phenotypes. That's the output. That's what you see. That's what the cell is able to do. Okay? Now here's what you have to understand. It's constantly evolving. It's changing. Because what you're going to see is that the selective pressures that are put on a cancer cell evolve over time. So that what is going to be the rate limiting step for a cell to start proliferating initially is going to be very, very different than the survival of a cell later on. Okay? So for example, the environment that a tumor cell finds itself in initially is pretty conducive to growth. But over time, as the tumor grows, the cells on the inside are trapped. They lack access to the circulatory system, so they start starving. They're starved for glucose. They're starved for oxygen. You're now providing a different selective pressure now on a cell that's on the interior of a tumor than one of the very initial cells that started that tumor off. Different selective pressure. You're going to select for different adaptive mutations that are going to allow the cell to evade that then. Okay? That means those cells are now going to change the oncogene that they're addicted to. The oncogene that they had that initially allowed them to first proliferate, that's not what's sustaining them anymore. It's a different mutation. Okay? It's always changing. And what this means is, if you're looking at it from a drug perspective, which we're going to be doing now, we're going to go back, and we're not only going to look at, well, how do you generate the oncogene, but how do you treat a cancer that is basically addicted to that oncogene? Well, you're going to see examples of where we can develop drugs to go after the oncogene itself. Target the oncogene, take it out, okay? Well, that can work in some cases, can't work in others, and you'll see why. The other is go after the hallmark. That is, don't go after the oncogene, but go after one of the phenotypic properties that the cell has. Or take the drug and go after the enabling characteristic. So you're going to see examples of all of these. And what they're going to drive home is the following, is that to treat any particular type of cancer now, you really have to understand what's called the genetic signature of that cancer at that time. It's a moving target, and you're going to have to be aware of that, especially in an oncology's context, to where you're having to stay one step ahead of where the tumor is going in terms of anticipating what sort of mutations it's acquired and where it's going to go. And you'll see examples of that as soon as we get into it. Okay? So, that's just a little framework to get you started. All right, so if we go up to the very top, all we're showing up there in the RTK pathway are some of the very initial events and the initial components. So what you guys can see is we start with the growth factor receptors that are RTKs. All right? We can target those. So what you see up there are some drugs. Anything that you see that ends in MAB, M-A-B, is a monoclonal antibody. It's something that's going to work by binding to the extracellular domain of the receptor and compromise its function. The other thing you can do with RTKs, that you'll see, is we can target the tyrosine kinase domain. Those are tinibs, like gefitinib and erlotinib. If you ever see the word tinib, it means it's a tyrosine kinase inhibitor. And you're going to see that, well, it's easier to take out a tyrosine kinase specifically than serine threonine kinases. Okay? So two different strategies there. Then we move to RAS, and you notice up there, well, the problem with RAS is, as you're going to see, is that a lot of people have thought it's undruggable. Because the best way to describe RAS, it's a little greasy ball with nothing to bind or grab onto. So, makes it a challenge. But you're going to see, people are smart. We can come up with ways to try and take out RAS. Then we go to RAS, the kinase that's activated by RAS,
And now you're going to see there's two approaches there. One is to directly go after and take out the kinase, and that's the drug vamurafenib. The other way you can do it is take a new approach with a drug that disrupts the interaction between RAS and RAF. Okay? And what you're going to see is that every one of these approaches works initially. And the reason they only work initially, because of all this stuff going on down here. So that the minute you start treating a cancer with one of these drugs, you're now applying a selective pressure. It is no different than if you're trying to kill a bacterial infection with an antibiotic. Of course, student health tries to kill everything with antibiotics. And as some of you know, it doesn't work very well. But what happens with antibiotic resistance is, as soon as you treat a bacterium with it, they go to town. And all you're doing is making them mad. Same thing happens with cancer cells. Treat them with a chemotherapeutic drug, you're just ticking them off. And all you're doing is providing an environment for the acquisition of adaptive mutations that allow that cell to survive. It's very slow. It's very inefficient. Maybe 1 out of 1,000,000 cells will acquire that adaptive mutation that allows it to grow. That's why what you're going to see, cancer progression is generally very slow. It takes a long time to select for a cell that acquires a mutation that allows it to bypass the drug, because more often than not, what you're going to see, the further upstream you initially target an oncogene, the more likely it is you're going to get an adaptive mutation, because all you have to do is change something that works downstream of that, and now your drug's no longer effective. Okay? So targeting the receptor is the first place to go. But if you become resistant to one of those drugs that targets the receptor by, for example, generating RAS or RAF oncogenes, well, targeting the receptor doesn't work anymore because the tumor cell doesn't need it, because now it's being addicted or driven by RAS or RAF, which are downstream. Okay? All right. So with that cheery intro, how can we take RTKs and turn them to oncogenes? There's many, many ways you can do this. So please understand, we're only skimming the surface here. We're only taking a look at a couple of prototypical examples. Kind of the main ones that are out there, the most prevalent ones. But there's lots of ways you can do it. Okay? So let's start off with HER2. There's HER2. Hangs out all by itself. Nothing really special about it. Except remember, there's something unusual about HER2. And that is HER2 doesn't normally bind to a ligand. Okay? So it's a ligand-independent RTK to begin with. And it only causes problems if you overexpress it, because it can homodimerize or it can form heterodimers with HER1, HER3, HER4. So normal levels of HER2 don't do anything. Okay? What happens, though, if it undergoes a point mutation? This is the other thing I want to go back to. It's a recurring theme you guys have seen all fall. The mutations that are involved here are subtle. Single nucleotide changes, single amino acid changes. We've gone through a litany of different diseases that arise from that. C to T change, progeria. You're going to see the same thing with many of these oncogenes. It doesn't take a huge genetic alteration to turn a well-behaved proto-oncogene into an oncogene. Okay, so what happens if we take HER2? Let's go down to the membrane-spanning alpha helix. It's embedded in the plasma membrane, phospholipid bilayer, and we've got a valine. Nice hydrophobic amino acid, just exactly what you'd expect to find in a membrane-spanning alpha helix. Hydrophobic, should be there. Well, look what happens if it undergoes a single nucleotide change, and that valine codon now changes to glutamine. Well, you're immediately thinking, it's polar. Heck with the polar, that ain't what's important. Glutamine, remember, the R group on a glutamine is an amide. It's got a carbonyl oxygen, it's got an amino proton. It can be a hydrogen bond donor and a hydrogen bond acceptor. And that's exactly what the oncogenic form here does.
and that is by changing that valine to glutamine. Now your HER2 receptors up there in the plasma membrane, even at low levels, can come together and hook up. And the way that they can pair up is you can form 2 hydrogen bonds between them within the membrane-spanning alpha helix that you could not form before. You just made an oncogene. The oncogene is referred to as new, the new oncogene, NEU. And the reason is that this particular mutant form of the HER2 receptor was discovered in a particular type of cancer called neuroblastoma. Okay? So oncogenes were more often as not named after the tumors that they give rise to. So this particular oncogenic version of HER2 acquired the name NEU or new simply because it was discovered in neuroblastoma. It's also one of the drivers involved in metastatic breast cancer. Okay? So there you go. These guys are always dimerized, even at low concentrations. It already doesn't need a ligand to bind to. You're off at the races. Those cells will proliferate in the absence of any growth factor. There's another way we can do it. Now, this is a genetic alteration that's a little bit more profound, and this affects HER1. Okay? HER1 is the most prevalent canonical EGF receptor that we have. Okay? It's normally well behaved, it doesn't dimerize unless it binds EGF. Everything's good about it. But in this one instance, it turned out it underwent a deletion. The very beginning of the gene actually was deleted. There was a chromosomal deletion of the first part of the HER2 gene that basically took out almost the entire extracellular domain. Remember, these are type 1 integral membrane proteins. So the N-terminus is here, the carboxy terminus is here. So it had a deletion that took out basically the front half of the protein. So translation would start at a methionine that happens to be down here. So we just have a little stub of the protein outside the cell. You would think, well, you took off the ligand binding domain. This thing ought to be dead as a doornail. It shouldn't be able to do anything, right? It can't bind a growth factor anymore. Well, no. The problem is what you actually removed wasn't just the ligand binding domain, but you actually removed this region here, which is important in keeping 3 monomeric receptors apart. It's a region of the receptor that pushes them apart from each other unless each of them is bound to a growth factor. That's the problem you create here. You delete that extracellular domain, and now what happens is you now have this little version here with very little outside the cell, but it's a constitutively active tyrosine kinase. And this was discovered in an unusual form of leukemia. Most leukemias that you're used to thinking about deal with the abnormal proliferation of immature white blood cells. This one is involved in the abnormal proliferation of stem cells, erythroblasts, that normally then subsequently differentiate into non-proliferative erythrocytes, immature red blood cells. The disease is called erythroblastosis. Okay? So, this was the altered receptor that was found in these cells. So this version of the HER1 receptor was given the name ERB-B, E-R-B-B. And that's because it was the oncogene that was the causative agent of erythroblastosis. Okay? So again, just understand the name given to the oncogene has nothing to do with the proto-oncogene's function. It only had to do with the tumor type that that gene was discovered in. Okay? All right, so here's the most important and the most prevalent mutation, however, that you find. And it doesn't have to do with changing the function of HER2. It has to do with changing the level of HER2. Okay? And this is most relevant to metastatic breast cancer. All right? And what it is, it's a process known as gene amplification. Normally, all of our DNA sequences are replicated once and only once per cell cycle. Certain genes have a proclivity to be replicated more than once. Cyclin D can do this, MYC can do it, and HER2 can do it. And what it means is that in these cells, during the course of normal DNA replication, the cell will go back and re-replicate a given gene more than once. You're amplifying it. You're making more copies. And this happens very frequently in metastatic breast cancer cells to the HER2 gene. So if you basically take a look, so here's a normal breast cell, and you're taking a look by staining chromosomes with fluorescent markers that tag the gene. So these two bright spots here are the nucleoli. And that's the sites of ribosomal RNA production, ribosome assembly, okay? Normal cells have two nucleoli. So they're a nice cytological marker to just look at a cell and you go, okay, there's two. Now, if you take a look in the same cell, these lesser staining objects that you're seeing, those are the HER2 genes, okay? So we all start off with two copies of HER2. One came from mom, one came from dad. So we're diploid. So a normal cell has two copies of HER2. There they are. And that's a normal cell. If you take a look, oftentimes in a metastatic breast cancer cell, so you do a biopsy, you take a look at it. And now what you notice is we still have two nucleoli. They're still there. But now what you see is 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 copies of HER2. What it means is that cell is going to be producing significantly more HER2 than the normal diploid cell. It's no longer diploid for HER2. So it's going to be overexpressing normal HER2. And what do you guys already know about HER2? What can HER2 do that none of the other EGF receptors can do? It can homodimerize and transmit signaling without binding to a growth factor. So what happens is you amplify the gene. Now you've got cells that are doing two things. They can proliferate without binding to any growth factor, but the other thing it does is, remember, HER2 can also form heterodimers with the other HERs, 1, 3, and 4. That means that you can also cause them to transduce signaling, even under conditions where you have very, very low EGF levels outside the cell. So these cells are going to proliferate in conditions that normal cells won't, even when there's vanishingly little EGF, or if there's none. So how do you go after it? Well, here was one of the very, very, very first examples of trying to take advantage of the fact that here's a tumor that has a unique genetic signature. And that is, these cells are overexpressing HER2. Normal cells don't. So if you could go in and target HER2, could you stop proliferation? That was the rationale. So they designed a monoclonal antibody that binds specifically to the extracellular domain of HER2. They called the drug Herceptin. It basically intercepts HER2. So what the monoclonal antibody does is it binds to the extracellular region of HER2. Now that's an advantage because it means you're dealing with a drug that does not have to get into the cell. It works outside the cell. It binds to the receptor and it does two things. The first thing it will do is it prevents EGF from binding. So any of the HER2s that have formed heterodimers with HER1, HER3, HER4, you're taking them out of action because they can no longer bind to the growth factor. But the other thing that it does is it causes the receptor to be desensitized. And so what it will do is that when Herceptin binds to these receptors, we start clearing them off of the plasma membrane. They get endocytosed very rapidly. So in essence, what you're doing is that you're causing the cells to stop proliferating by clearing out
HER2 receptors from the plasma membrane. Okay? The nice thing is it's very specific. Doesn't affect cells that don't have overexpressed HER2. So we're not targeting those. Now, what's a drawback here? Or what's a limitation here? Yeah.
Only work as long as you take the drug.
They're all, yeah, this is true. Why is that?
You take the drug and it back to overexpressing.
Okay, so what does that mean the drug is doing or not doing? It's not changing the gene. What else? Are you killing the cells? No, you're not killing them. All you're doing is removing the signal for them to proliferate. Okay? But you're not killing them. So generally, Herceptin only works as an adjunct than to do a co-treatment with a cytotoxic drug like Taxol. Okay? So it's a combinatorial therapy. So that an individual with metastatic breast cancer, what you find, will be taking Herceptin. That stops the cells from proliferating, but then you also have to treat the person with Taxol to kill those cells. What's the problem with that now? There's no specificity for the Taxol. You get all the horrific side effects, anemia, hair loss. All of those are because Taxol is going to kill any proliferative cell. It doesn't know if it's in a cancer cell or a normal cell. So the problem with a drug like Taxol or any sort of cytotoxic agent is how do we target it to just the tumor cell? This is how Herceptin is now evolved. Herceptin is going to allow you to generate what's called a smart bomb. And here's what you do. You take Herceptin, okay? And those of you that are organic chemists can immediately discern what we've done here, is that we've added a little linker here. And what the linker has is a thioether bond, which happens to be labile to an acidic pH that you're going to find in the lysosome. And what you do is you hook up to Herceptin, the antibody, is that we've hooked up a cytotoxic drug. In this case, the drug is a compound called DM1 or mitacine. And what it is it's a drug that actually causes microtubule depolymerization. Okay? Cytotoxic does all get out. But look what you've done to it. You've now turned Herceptin from a blocking antibody to a lethal killer. And the way that you've done it now is that the Herceptin here is conjugated with, say, 3 or 4 molecules of DM1. And now what happens is this form of Herceptin can now bind to the HER2, just like it did. It gets endocytosed, so you're knocking down the levels of the receptor. But what happens is it comes in, it basically goes and gets to the lysosome, and that's where that thioether bond gets cleaved. And now it releases DM1, which then goes out and causes all the microtubules to depolymerize. Well, there goes your vesicle trafficking. There goes the ability to make a mitotic spindle. Cell dies. Look what you did. You just turned a generic cytotoxic drug into a smart bomb because this is your delivery device. What are the potential applications of this? The same phenomenon takes place with any integral membrane protein. Any integral membrane protein that you can find on a cell, that when it binds to an extracellular antibody, will be endocytosed. It's just sort of a natural feedback mechanism all cells have. So the applicability of this approach is that if you can identify on a tumor cell a unique integral membrane protein, and you can develop an antibody that will recognize the extracellular domain of that protein, conjugate it to a toxin, there's your delivery device to target that drug just to the tumor cell.
And this was an approach that John Hare was working on in his laboratory for the last several years before he passed away earlier this fall with a heart attack. But John was basically taking this approach, and that is try and search and find on any tumor cell that's out there, find a unique integral membrane protein, and now what you have is your gateway to get a cytotoxic drug into the cell. Okay? So it's a very powerful approach, and it's one that you're seeing used increasingly. The problem with chemotherapy and cancer, as you guys are going to see, it's in the delivery. Delivery is the rate-limiting step. There's toxins all over the place. We've got enough stuff out there to kill any cell on demand. That isn't the problem. The problem is getting them delivered just to the cancer cells. So that way it's specific, and you avoid all of the collateral damage, which we call side effects. Okay? All right, so there you go. Now you guys can see how we can target at the level of a receptor. But like all good things, it doesn't work forever. So here's how we roll down the road here of adaptive mutations. Okay, metastatic colon cancer is often driven by HER1 having some sort of a mutation or being overexpressed. So one of the most common ways to initially block the proliferation of metastatic colon cancer cells is to use an antibody. It's sort of like Herceptin, and it's called cetuximab. Cetuximab binds to EGF receptors that are overexpressed on colorectal cells, stops their proliferation. Works very, very, very well. It will stop the proliferation of the tumor. Oftentimes it can then be surgically removed at a very, very, very early stage, and you would think that you're in remission. Not so fast. So here's what's happening, is that initially an individual has a tumor that's being treated with cetuximab. Tumor goes into remission, nothing's happening. Look at the time frame here. The individual is basically being treated for weeks, okay, with cetuximab. So 16 weeks, call it 4 months out. During that time, no further tumor progression. But now what happens is you start noticing here, a person goes back in for a CAT scan or an MRI, and what you now notice is that the tumors return. It's growing again. Treatment quit working, and in fact, it's because the tumor is now resistant to cetuximab. What's happened? Well, when you treated the tumor with cetuximab, you're putting selective pressure on the cells. And you're putting selective pressure on them to acquire an oncogene that functions downstream of the EGF receptor. It takes time. It takes about 4 months for this to happen. And it turns out that more often as not, the oncogene that you're now selecting for is going to be RAS, and to a lesser extent, RAF. Now here's something that you can find, though, diagnostically. If all you were doing was following the tumor by imaging, the tumor would have already resurrected and started growing again very late, after you've already, in order to see it. However, one of the things that tumor cells do is that they release DNA into the bloodstream. All cells do it to a certain extent, but tumor cells do it to a very large extent. It's not clear if it's just because they're constantly dying and releasing DNA into the bloodstream, or if they actually have a ramped up secretory pathway and DNA gets secreted into the bloodstream. But the point is, you can actually monitor what's going on in a person being treated with a chemotherapeutic drug by pricking their finger and just doing a DNA analysis from their blood. And if you do that in this case, here's what you would have found. Is that going as early as 4 weeks into the treatment, a month or 2 months earlier, what you would start seeing is that if you're analyzing the DNA in the bloodstream of that patient, they've acquired an oncogenic mutation in RAS. It's already starting. You've got a new cohort of cells that have acquired an adaptive mutation, and as long as you keep treating them with cetuximab, you're selecting for these cells. They're already starting to proliferate. But what it means is this. If you would have waited to image by a CAT scan or an MRI the tumor and show that it was growing again, you're having to wait 4 months to see that. You do the DNA analysis, you could find it as earlier as 2 months earlier than that. What does it mean? Well, it means that what you would do, you'd start taking the patient off of cetuximab and you would start treating them with something else. Because what it means is you now have an emerging cohort of cells that are no longer going to respond to cetuximab. Because now they're addicted to a new oncogene. They're not addicted to HER1 anymore. They're addicted to RAS or RAF. So now what you're finding is that this sort of genetic analysis is mainstream in oncology. This is being done now routinely for any individual being treated for any type of cancer. And what you're doing is you're genotyping and seeing what mutations are these individuals acquiring that you can just measure in the blood, doesn't require a biopsy, and you can actually see it's time to change the treatment earlier than it would have been because now they've acquired a new mutation. Okay? So now we're at RAS. So this is actually, I just threw this little slide up here. I gave it to you guys separately because it just came out this week. So it's kind of a cool little slide. So here, let's just add. It's better than the ones in the textbook. Here's the RAS cycle. RAS is a GTPase. You already know that RAS has to interact with SOS, which is interacting with the receptor. That's the GEF. So RAS has to go through a cycle of interacting with SOS to exchange GDP for GTP, and that turns it on. Okay? Now, RAS has a gap. And the gap for RAS is a tumor suppressor called NF1, neurofibromatosis 1. The normal function of NF1 is to cause RAS to hydrolyze that GTP very quickly and shut itself off. And that means it has to go back, bind to SOS again, pop out the GDP, get a fresh GTP. So RAS has to go through this cycle repeatedly for normal proliferation. Well, it can jump out of this cycle or bypass it 2 ways. One of them is that if we take out NF1, if we lose the tumor suppressor. Well, if we take out NF1, we're taking this out of the pathway. We're taking the gap. So what it means is RAS will be hyperactive in the absence of NF1. It can still hydrolyze GTP, but it does it about 1,000 times slower. So it means RAS is going to be propagating a signal for a longer duration than it normally would. But that's not the kicker. When you're acquiring adaptive mutations that are causing cells in the previous case to proliferate in the absence of the HER1 receptor, they're not going after NF1. The mutation is in RAS. And what you're going to see is that it is called RAS-D, superscript D for dominant. And that's because these RAS oncogenes can never hydrolyze GTP. They lack the GTPase activity. They're constitutively active. So now what that means is RAS is always on. That means you don't need the receptor, you don't need SOS, we don't need a growth factor. We've moved downstream of that. Okay? If you take a look, this is a listing of all the prevalent mutations that are acquired in individuals who have become resistant to EGFR treatments. So these are individuals that would be resistant to cetuximab. They would be resistant to Herceptin, for example. Here's what you find. You notice that all of these guys here are downstream after the receptor. Okay? We have 3 RAS alleles. Or not alleles, genes. We have 3 different RAS genes. KRAS, HRAS, and NRAS. NRAS is typically restricted to neural stem cells. So most of the culprits that are going to be converted to oncogenes in non-neuronal cells are KRAS and HRAS. take a look and see where most of them fall. If you take a look in tumors that have become resistant to treating with EGF blockers, here's where most of them are. 40% of them are within the RAS gene. What's been affected? One of two amino acids, either glycine 11 or glycine 12. And if you take a look at the pie chart here, now you can also see that basically you can also acquire other mutations in RAS, but also some of the downstream components like RAF. But this is the majority of them. So the vast majority of time, when you acquire resistance to a drug that's targeting the receptor, you're going to have a mutation in RAS. And the RAS mutation is going to knock out its ability to hydrolyze GTP. Here's why. If we take a look at RAS, the structure of RAS, here's GTP. Here's alpha, beta, there's the gamma phosphate. This is the one that the RAS GTPase can normally cleave and shut itself off. And NF1 accelerates that. Here's glycine 12. Here's glutamine 61. They are poised right next to that gamma phosphate. And here's what happens. If you change either glycine 12 or glutamine 61, they are the mutational hotspots in RAS. Any mutation that changes either of those amino acids to anything else, I mean anything else, RAS can no longer hydrolyze GTP. It makes sense because glycine is the smallest amino out there. It's the smallest amino acid out there. It's got a proton as its R group. Even if you put alanine there, it's got a methyl group. Put any other amino acid there, it's got a much, much bigger R group, and that's basically going to shield that gamma phosphate, and RAS can no longer hydrolyze it. So it turns out all of these amino acids up here, they're the mutational hotspots for RAS. Change any one of them to any other amino acid, RAS is now constitutively active. It's always going to be active, even in the absence of a GEF, in the absence of a growth factor. The signal is now being propagated downstream. Herceptin's now no longer effective. Cetuximab's no longer effective. How frequently does this happen? A lot. So if you take a look now, and you go tumor by tumor by tumor, and you take a look at these different tumor types, and you say, what proportion of these tumors have an oncogenic form of RAS? A lot of them. Especially if you take a look at pancreatic cancer. Tops the list. 90% of pancreatic cancer, which is pretty fatal because it's so rapidly aggressive, there's no real treatment that works. 90% of them have an oncogenic form of RAS. Okay? So this isn't an oddball, this isn't a rare exception, this is the norm. The RAS oncogene generally comes about in the vast majority of human cancers. It's the single most prevalent oncogene you will find present in most human cancer cells. Okay? Well, so here's the problem. Is that up until very recently, it's undruggable. No one's been able to find a drug that can be used to treat RAS oncogenes. Think about it. The reason the RAS is an oncogene is because it can't do something. That's the dilemma. The reason RAS becomes an oncogene is because it cannot hydrolyze GTP. Most drugs inhibit something. Well, you can't inhibit something that it's not doing. That's the dilemma. What makes it more complicated is that RAS is a greasy, slimy little ball globular protein with nothing that a drug can bind to. So for many, many years, oncologists have basically said, to hell with it, we can't take this thing out. Well, then Hal Varmus, who was the director of the NIH, said, I don't buy this. Everything's druggable, okay? You just need to work at it, okay? So people have. So here's some strategies to attack RAS, okay? Just to kind of go through what you might be able to do. Okay, well, you got here, this is trying to basically get at the proteins that interact with RAS. We'll pick it up there on Monday. Come down here. In some instances, glycine 12 has been changed to cysteine. Well, the nice thing about cysteine, it's a very reactive amino acid. It's got a nice sulfhydryl group, sulfur. You can target compounds that can bind to sulfur and can perturb the structure of the protein. So there's one. You can take advantage of that specific amino acid substitution, and that might be effective for about 20% of lung cancers. The other thing to do is to try and create a pocket that another drug can bind to, kind of pry open the structure of RAS so that something can bind to it. And then up here's the one I just want to finish up with today. And that is, remember, RAS is lipid anchored. So could you come up with a drug that would prevent RAS from being anchored to the cytosolic leaflet of the plasma membrane? And would it work? And the answer was yes, and then no. And it also illustrates how cancer cells will do whatever it takes to survive against what you hit them with. Here's the drug. Remember, we just talked about it a week or two ago. Lenarfinib. Remember? Lenarfinib is a farnesyltransferase inhibitor. So here's RAS. RAS has to go through the same process that lamin A does. And only the difference here is RAS keeps that lipid anchor. So the farnesyltransferase catalyzes the first step that covalently attaches farnesylate to the cysteine near the end of RAS. And that's normally necessary for RAS to work its way up to the cytosolic leaflet to anchor it there so it can be activated by SOS, and then it can also activate RAF. Cool. Lenarfinib keeps that from happening. So remember, lenarfinib was actually first developed as a chemotherapeutic drug. You take lenarfinib, treat cells in culture that are being driven by a RAS oncogene, they stop proliferating. And you take a look at them, and all the RAS is falling out of the plasma membrane. It's not there anymore. Looks great. Well, that's in the short run. So now you move to clinical trials. And so you started treating individuals with cancer using lenarfinib. It worked initially. Maybe a few weeks, sort of like what you saw with cetuximab. But then the cells become resistant. And here's how they become resistant. The cells have a backup mechanism to put RAS into the plasma membrane. So what happens is, if you inhibit the farnesyltransferase with lenarfinib, okay, you keep that. Well, give the cells time, and they just switch over to a backup program. And the backup program is that there's another lipid over here called geranyl genarl. Does the same thing. It's a different transferase. That transferase is not inhibited by lenarfinib. So what happens is you slow the cancer cells down. They get angry. They get ticked off. They just come over here, and now they will start attaching this lipid anchor, go right back up to the plasma membrane, and they keep right on trucking like you didn't do anything to them at all. So this illustrates how cells will switch from one mechanism to a backup mechanism to accomplish the same end because of this treatment here. So this just sort of illustrates one of the pitfalls of targeting any oncogene. Okay? All right. So on Monday, we'll pick it up, and we'll just keep going through with some more examples. And I've got some more slides and everything to put up for the rest of the cancer group.